Hello, good morning, and welcome to the International Plant Breeding Seminar Series. This is the 5th seminar for this series, and we are hosting it from NC State University here in North Carolina. We have the pleasure today to have Dr. Lukas Mueller from Boyce Thompson Institute presenting about Breedbase. Lukas got a degree in biology from ETH in Switzerland, and then a PhD from Lausanne University in biochemistry. He also did a postdoc at Stanford University before joining Cornell University in 2003, and later on the Boyce Thompson Institute, where he is a professor there. Lukas' lab is in charge of, has developed a lot of databases that have helped a lot of breeders, particularly in less research crops. I know Lukas from his work within next-gen cassava, but I know he has developed databases for yams, sweet potato, Solanaceae, Musa, and goes on and on. I think it's an extremely helpful work that his lab does for all of us in the world. And it's a pleasure to have you here with us, Lukas. I'm going to stop sharing the screen and give you the floor to you, okay?
Yeah, thank you very much. Can you hear me?
Yes, maybe a bit closer to the mic.
Hello, hello.
Yeah,
Is this better?
I think it's good.
Great. And I just shared the screen, so I hope you can see that.
Yes, we can see it. Yeah.
And so today I'm talking about Breedbase. Thank you for the kind introduction, by the way. And Breedbase is basically a system that helps breeders managing their breeding program, and it really does that by implementing what we call this digital ecosystem for plant breeding. And I just want to note that this crazy picture that you see here is made by DALL-E, artificial intelligence software, and I have no idea why I put the smokestacks in the background. So I'm at the Boyce Thompson Institute in Ithaca, New York. It's a small plant biology institute. It has about 15 research groups. We just have a new president, Silvia Restrepo. Everybody's very excited. And if you want to know more about the institute, there is the URL, btiscience.org. So this work, as Carlos just mentioned, started really out with cassava, and we were asked to create a breeding database for this next-gen cassava project, and the database we created was CassavaBase at cassavabase.org. And cassava is a very interesting plant, and let me just say a few words about cassava and about the other RTB crops that we have been working on, because I think not many people may know so much about these, as they're, for many people, a little bit unusual crops. Cassava is a tropical and subtropical plant that's mainly grown for its starchy roots. Interestingly, it is actually native to South America, but is now really important in Africa. And you see here how it's prepared and all the different dishes that can be prepared, and the pictures of the roots. The leaves also are sometimes eaten and have this interesting shape. And so estimates vary from, between 500 and 1 billion people that get their calories from cassava. And that's really, up there with, or just after rice, maize, and wheat, and up there with potato. And the Next Gen Cassava project had this idea to use genomic selection to improve gain and speed up the breeding cycle for cassava. And we have worked a lot with these cassava breeders, and I think I just want to give a shout out to all these people who, from the early days of Breedbase, have contributed a lot of feedback, and has been really extremely nice to work with, like Chidozi, Ismail, Robert, Enrico, Hernan, Xiaofei, and many others, and of course many in their staff as well. So that has been fantastic. As you'll see, one interesting fact about cassava is that it's actually toxic, and it needs to be heated so that the cyanide goes away. What's happening is that the roots accumulate linamarin, and linamarin then essentially decays into cyanide, which is of course very toxic, and so it has to be processed before it can be eaten, and I found that quite unusual for a food crop. Some other crops we've been working on, and Carlos has mentioned that, is sweet potato, Ipomeo batatas, which of course is not directly related to potato, but is in the family of the Convolvulaceae. It's also a New World crop. It's also clonally propagated like cassava. What makes it difficult to breed is that it is a hexaploid genome, so it's very, very complex. Also, a nice factoid is that some sweet potato breeders won the World Food Prize in 2016, Maria Andrade, Jan Lowe, and Robert Manga. And of course, we work with them and also with Craig Yancho, who is the US sweet potato breeder in NCSU. So, and so there's one big problem with sweet potato in the US, that people confuse it with yam. And of course, yam is the other one that we work with in another project called Africa Yam. Sweet potato and yam couldn't be further apart almost botanically. Yam is a monocot. It is native to West Africa and Southeast Asia. There are different species that are being used. One little curveball it throws at the breeders is that it's a dioecious plant, so there are female and male plants, but it's a very, very high value crop. The roots are worth a lot, and again, it's not sweet potato. And again, we have had very fruitful projects with Azrad and Agre and Jude and all the other breeders and their staff. And finally, we have banana. Banana is maybe the most complex plant to breed. We are talking here about cooking bananas. They are different from the Cavendish banana that you can buy at the supermarket. We are talking about Eastern Highland bananas that are the main types are Matoke bananas from Uganda, which you prepare a meal from called matoke, which is very nice. It's steamed bananas in a banana leaf. It's very tasty. And the Amchare bananas from Tanzania, and also plantain that's more grown in Western Africa. And so if you have recently eaten a banana, you might have realized there is no seed in a banana. So that makes it very hard to do any kind of breeding, and it would take too long to explain the breeding process here. But the short answer is the triploids usually don't have seed. There are diploids and tetraploid progenitors that are being used for breeding, and then a triploid is being created by crossing a diploid to a tetraploid, and that sounds pretty crazy to me still, but that's how it works. And you have to be really fearless to breed bananas, and we have again had very good collaboration with Ronnie, Brigitte, Ellen, and all their staff. So we have created all these databases. So cassava base I mentioned. Actually, there was a database before that called SGN, soilgenomics.net, which of course still exists and is a database for tomato, potato, and other Solanaceae. And so this is kind of a phylogenetic tree of databases. And you see that from cassava base, we then created these other databases. And of course, now a lot of people are using Breedbase for their breeding needs. And of course, this tree would be very, very large if I showed it in its entirety, what other people have created using Breedbase. So why do we need databases at all? I mean, breeding was done for a long time without any kind of database, just lab books. And so I found this interesting overview in a paper by Ed Buckler that was recently published in 2018, and it really gives you the different eras of breeding. So breeding 1.0 would be more, incidental selection by farmers. So during farming, you of course also select things, and that's how things have been improved over thousands of years, of course. Breeding 2.0 is when you start to use more defined experimental designs, and you are systematically selecting the best lines. Breeding 3.0 is where we are now, and what you see here is that we start to use genomic data. And when you start to do that, when you start to fly drones, as it's shown here, or you use genotypic data, that's just a massive amount of data that you need to manage really well, because at the end, of course, all the data has to match together and has to be useful, right? Breeding 4.0 is then, of course, when you make designer genomes using CRISPR and things like that. We are not quite there yet, I think, but that will happen soon too. And of course, you need vast amounts of data for that as well. This is just a nice picture from some of the more historical breeding books of IITA. Those are cassava books. And you see that there's a lot of information in these books, and they're all being digitized now, or some of them at least, and added to cassava base. But you see the problem of this approach is, how do you calculate on these data, and how do you really, from these books, efficiently create breeding decisions? And of course, people started using Excel, but Excel also has big limitations. So now what these breeding data looks like is like this. This is the server room downstairs, the BTI, where we run all these databases. And of course, they don't look as nice as the books, but it's much more efficient to create statistical analysis from the data than with the books. So it's much more efficient. Just to give you an idea how much data a database like cassava base contains, we have thousands of trials in the database. Currently, I think about over 6,000. We have over 5 million stock entries. Those include plant plots, tissue sample entries, and we have almost 25 million trait measurements. So these databases can be massive, and these databases really scale to that level of data. Another kind of impressive view in the cassava base database is all the breeding locations that people have added to the database. And you see that they're spanning the entire globe, and that there are a lot of locations. I think it's just quite impressive to see this wide use of the database. So when I'm talking about cassava base and the other breed bases, they're extremely easy to use because they're all web-based. You don't have to install anything. You can just use a web browser and you can connect to them. Of course, you need a login, but then everything is web-based and has nice web interfaces. So if you know how to use the web, you essentially know how to use this software. Of course, there is some complexity associated with using it, but essentially it's just web paradigms that are being used. So I want to briefly talk about phenotyping that we are doing in cassava base and Breedbase, and that is supported by the database. And before you can do that, actually, of course, you need to have a concept of a field in the database. The database can generate field randomizations for you. You can view these randomizations, they're stored in the database. You can download them to a tablet or other device, and you can do phenotyping, and a lot of different randomizations are supported, standard randomizations that all breeders are using. And I just want to give a shout out to Alex, who created part of this tool here. Then another important part is that you need to barcode your fields, because if you don't barcode the field, your data quality will be much worse. So when you go to a plot or a plant, you just want to scan the label and then do your phenotyping. And Breedbase supports this barcoding. It has its own label designer. You can design labels. You can put 2D barcodes on it, or 1D barcodes, and so forth. And so that's really a key step to do to increase your data quality. Then there's different ways to collect data from these fields. I think the most important is the Pheno app called Fieldbook, which is downloadable from the Google Store. Of course, there are other ways. For example, a lot of people start using drones, and Breedbase has a drone component that we'll talk about in a moment. And also people are starting, because Breedbase deals so much with root crops, people are starting to use ground penetrating radar to look at the roots as they're growing in the field non-destructively. And this is not supported yet, but it's something we are working on, and IITA is working on, and hopefully we can support that in the very near future. What's really important recently is the NIRS data, the near infrared spectra that you can take in the field. There are new devices out that are portable and that really revolutionize how phenotyping is done in the field. I just want to say a few words about the Fieldbook because it is so important. We work very closely with Trevor Reif at Clemson to integrate the workflows of the Fieldbook with Breedbase. It's very easy to install it. It's a free install from the Google App Store. And then, these workflows that we've created are really a seamless data exchange with Breedbase through a technology called BRAPI. And the Pheno apps comprise some other tools that I'll mention. There are some other apps that we fully support, and I will mention them at a later point. Yeah, the nearest phenotyping has really revolutionized phenotyping in the field because these spectra can actually be correlated with properties and sometimes specific compounds or other phenotypes such as dry matter. And essentially, you have to just correlate the spectra with these properties, so you can generate calibration curves, and with one scan in the field, you can, for example, determine the dry weight of a sample. And so what you can do in the Breedbase is you can store these spectra, but you can also go through this entire calibration routine. So you can create essentially new traits that you can measure with a near spectrum. And you can then also, from a spectrum, you can predict those traits all within Breedbase. So that's a very important functionality. Another important thing is drone data collection. And again, Breedbase can support some workflows for drone imagery. For example, you can assemble drone images into these complete field images. You can then overlay the plots on these images, and you can excise each plot, and you can have different wavelengths and so forth for the different plots. And you can run then analysis on these different plots and derive traits from them, such as growth and so forth. But just a brief word about genotyping. What's important, of course, for genomic selection approaches is that you need a complete set of genotypes in the database. Breedbase stores genotyping data and phenotyping data all in the same database, and a lot of other data such as field data, soil data, and you can also be linked to weather data. So you get everything in one database, and that really allows you, as you can imagine, to very simply apply a lot of algorithms to all these data. And so genotyping workflows are very complex, actually, because you need to go to the field, you need to collect the samples, you need to extract them. You have things in a plate, which you need to transfer to a sequencing facility. And then the sequencing facility will give you some data back. It might be not exactly the data that you can use, but other data. So you need to process that data. You may need to impute that data, and you need to load the data back into the database. And so Breedbase has, for most of these steps, has very good standard operating procedures that guarantee a high data quality for your genotyping data. It's really important that the genotyping data is correctly assigned, obviously, to a certain genotype or stock. If you don't get that right, your whole predictions obviously will be wrong. So there's a high level of effort to make sure that this is correct. And again, you need barcoded fields for sample collection and so forth. And again, there's an app for that in the Pheno app that Breedbase integrates with, which is called Coordinate app. And what the Coordinate app does is you can take it and you can scan the barcode in the field for a line that you would like to genotype. You can assign a position in a 96 well plate, and you can then store that sample in that 96 well plate, extract it, and do whatever you need. The Coordinate app will communicate with Breedbase and upload all the information to Breedbase, what each sample is and what each accession is and so forth. And Breedbase can then submit this information to certain genotyping centers. And sometimes you just have to download it, an order form that exists for certain genotyping vendors, and some support even fully automated workflow, again, using the BRAPI technology. Genotypes are also very voluminous, and it's very hard to store them, actually. So we have developed special techniques to store them directly in Breedbase so that we always have all the data together, all the phenotypic data and the genotypic data. And there's a paper about it if you want to know more about it, how we store the high density genotyping data in the Breedbase database schema. Now, of course, having all this genotypic data and phenotypic data in one database makes it very easy to do things where you correlate phenotypic and genotypic data, such as genomic selection. Another application is, for example, GWAS. That's all very, very easy to do if you have all the data right there. And Breedbase supports these applications, but it has a wide variety of other tools. This is just the Analyze menu from the Cassava-based site. And you see that there is a lot of tools. You can analyze population structure. You can cluster things. You can look at kinship. You can look at stability of genotypes using AMI or GGE. You can look at the heritability of your lines and of your traits. You can run mixed models. You can compare trials. You can do graphical filtering and so forth. And as I mentioned, you can do, because you have the genotypic information and the phenotypic information right there at your fingertips. There is a GWAS tool that allows you to, run it and get Manhattan plots. There's also the genomic selection tool called SolGS that allows you to run an entire genomic selection workflow. And so the genomic selection workflow it implements has two parts. It has a model training cycle where you train the model. And then, so you need to have lines that are genotyped and phenotyped. And the line development cycle, you essentially can then predict phenotypes from genotypic information only. And of course, you can do that when the plant is very young. And that really compresses your breeding cycle time, which of course yields more improvement per unit of time. And this is what we call the tool. It's called SolGS. And you can do all what I just said. It also, has some analytical tools built in. You can verify the data. You want to make sure that how the data is distributed and so forth. You can also build selection indices, and then it makes predictions that you can use to advance the breeding cycle. And this tool was implemented by Isaac Tekle. We've recently added a new tool that we call the GPCP tool, which is a tool for genomic prediction of cross performance. So we don't predict how well a single line performs, but we predict which are the best crosses to make based on the predicted phenotypes in the based on genotypic models. So with this, in a sense, it starts with the same approach that we predict phenotypic data from genotypic data, but then we can also predict the best combinations based on these genotypic and phenotypic data. And then we rank the best combinations that are then suggested for crossing. It also uses a selection index. And this tool was created by a graduate student, Christine Yaga, with the help of some of the African yam partners like Agre, Azrad, Marley Labru, and also Marnin Wolfe, and of course, Isaac was involved in this tool as well. Then there are a lot of tools to visualize data. You can do PCA analysis, you can do graphical filtering, you can do correlations, you can look at distributions. And I think that's always very important to have this graphical feedback for breeders. I think breeders and biologists are usually very visual people, and that helps a lot in seeing what the data looks like. So what's very important also is that once you made your breeding decisions and you would like to go to the field and make the crosses, you need a powerful crossing manager. And crossing is actually a fairly complex workflow because you need to track a lot of stuff. You need to track the two parents, and you need to track offspring. You need to track, how many flowers were pollinated, how many fruits you got, how many seeds you got, and so forth. And so, as you might have guessed, there is an app for that from the PhenoApps group, which is the Intercross app, and that also works seamlessly with Breedbase. You can download wish lists to the Intercross app, what you would like to see crossed, and potentially that might not be possible because the plants are not ready at the right time. But also you don't want to make too many crosses of the same type, so it has limits of how many you should do, and it tells you when you have done enough crosses of a certain type, and so forth. When you then upload the data back into Breedbase creates the statistics for you, what you have crossed, and it creates it can create new seed lots, and it can create new accessions that you can then move onto fields and phenotype and start the cycle again. Breedbase also interfaces with another app called BTrack that is created by Trushar Shah and Margaret Karanja of IITA in Kenya. And this is a banana-specific app that is very complex. As we have heard, banana is always more complex than other species. And it can also track the in vitro regeneration of banana clones. And this tool was implemented by Tidima here in the lab. Of course, when you use the crossing tool, the pedigree will be built transparently in the background for you. You don't have to do anything, and your pedigrees will appear on a accession detail page like this. It's an interactive view where you can zoom around and you can expand your pedigree tree in different directions using these arrows. And this tool was implemented by David Lyon. There's also a module for inventory. You want to track, of course, your seed and your planting materials. For example, in yam, the planting materials, or in other clonal crops, the planting materials may not be seed. So you can track those as well. In the database, everything is barcoded, and there's a storage location, and of course the quantities are tracked of how much planting material or seed is available. And this tracking is done using transactions. So you can generate new seed lots as we have seen with the crossing tool, and then you can fill seed in other seed lots, or you can use them on a trial in a field, and so all these events are essentially stored as transactions, and how much how much you have taken out or added to a certain seed lot, and we'll give you the amount that is at hand in your inventory. You've also added recently an ordering system, so that these systems are usually multi-user, multi-breeding program systems, and you can set up some accounts that are actually vendors of clones and other items. And when you have this vendor role, you can then elevate stocks and other items to the so-called catalog. Other users can browse the catalog and can, if they find something interesting, they can click on it and generate an order that will send an order to the vendor. And the vendor sees the order, gets an email, and gets some status page where all the orders are listed, and can then start processing the order and set different statuses for the order until the order is completed and the person who ordered the item has received the order. Just a brief word how we develop the Breedbase software. We are loosely using an agile, so-called agile methodology, which means that we strive for maximum flexibility in the development process, because if you develop something, especially in an area like breeding, which is breeding technology is moving extremely fast. The new genotyping and phenotyping methods are coming up very quickly and are also very quickly outdated again. So the key is to be fast and flexible. And that's what the agile methodology provides. Everything is released under an MIT license, which means it's open source and everybody can use it. All the code is available on GitHub, and we deploy, since a couple of years, we use Docker, and we build new Dockers every other week or so. And we use a lot of open standards such as BRAPI. BRAPI is the breeding application programming interface that is trying to establish basically an open language for breeding data between computers. Another thing that is very important is that development of such a system has to be test-driven. So we have automated tests. That's a standard practice in software development. But it's just extremely important because these systems are very, very complex. And how do you know that one part stops working. So you need automated tests, and there are different types of tests. There are so-called unit tests, and there are so-called integration tests, so that you know that everything works in isolation that you program, but also if you put everything together, it still works. And so you want to run such tests, such automated tests really, really often. And this is all automated in the development process. There's a development workflow. And so these tests are run every time a pull request is generated, for example. So we always know what doesn't work. And of course, we don't merge anything that has a test that fails, and sometimes it's really hard to figure this out. So, but that's a basic tenet of the development process. Of course, tests never cover everything, and so there are usually test gaps. As we discover them, we of course try to fill them in. So the question is maybe how can I use Breedbase, or how can you use Breedbase? As I said, Breedbase is free and open source. Many people use it now for other species than cassava, yams, sweet potato, and banana that I've been talking about. But there are people who use it for potato, for rice, for wheat, for maize, sugarcane, diverse vegetables such as tomato and cucumbers, barley, oat, beans, and so forth. So it's almost any common crop may have already a Breedbase instance somewhere, and you can actually just go and clone this GitHub repository here. And when you do that, of course, you have to install Docker and Git. But when you do that and you clone this, you can just run it, and it will run 5 minutes later. So that's installing it is really, really easy. It has actually been downloaded more than 5,000 times, which is quite a lot for such a complex piece of software. We also try to have some hosting for people who cannot host it themselves. This is, of course, not free, but it's fairly professionally done, I hope. And if you would like to use that option, you can contact me at my email address. I would also mention that we have some upcoming events at the Plant and Animal Genome Conference. There is a Breedbase symposium where we will have a number of talks about Breedbase. I don't know what day exactly it is. I think in past PAGs it was on Tuesday, but that may change. And we also may have a booth this year that we share with BTI. So if you're at PAG, come and see the symposium, and come and see the booth. There are some resources available online. There's a manual online at this URL. It's also linked from all the Breedbase instances. There are some instructional videos on YouTube. You just have to search for Breedbase, and you'll see all the videos there. This is where we store all the code on GitHub, and this is where the system is stored on Docker Hub. I also want to mention that we this year published, no, it was last year, sorry. We published a paper about Breedbase, and if you're interested, you should probably look at that as well. And with that, I would just like to acknowledge people involved in creating Breedbase here at the Institute. The lab is actually not as big as here. There are lots of visitors and student interns here in this picture. And here to the left is actually Prasad, who was visiting at the time when we took the picture. And yeah, he has contributed a lot to Breedbase, so working for IITA. And with that, I thank you for your attention, and if you have any questions.
Thank you, Lucas. Thank you so much, not only for the presentation, but for all the work that you do supporting programs in not only root and tuber crops, but a lot of other crops. Please post your questions in the chat. We already have one comment and one question. A comment from Eric saying that Fieldbook app is fantastic. I would recommend it to anyone who has not used it. It's better than any proprietary data collection application that I have seen in my career. So it's a good comment about Fieldbook. The question is, what's the minimum hardware needed to host Breedbase on your own?
It depends on the size of your program and what you like to do. And if you, for example, have huge amounts of genotypic data that's very high density, then of course you need a lot of hardware infrastructure, a lot of disk, a lot of RAM. If you have a small breeding program and you mostly focus on phenotypic data, you could easily run it on your laptop with 8 GB of RAM, totally sufficient. But I would recommend going a little higher if you want to install it somewhere. Maybe 128 GB of RAM and 32 cores or something like that, and rated disks, a few dozens of terabytes at least. So fairly standard, and it runs on Linux in a Docker. So the Docker, of course, can be run anywhere. It can be run on a Mac, it can be run on Windows, but we just find that it runs best on Linux itself. For some reason, it's much faster. So I would recommend hosting it on a Linux server.
Okay. Emily is asking, we said there's a great talk, and she's interested in the GPCP tool for simulating crosses, and she was wondering if you might be able to describe a bit more how the simulation works. Yeah,
that's a good question. I'm probably not the best person to give this presentation. I haven't really prepared to talk about it. Christine might be a much better person to ask. But essentially what you can do is you have the genotypes for all your lines, and these are the genotypes that you would like to cross to each other, right? Then you can actually make predictions. You can it really what it does, it creates all possible combinations, right? And then it looks at what are the potential for each marker, right? What are the potential outcomes when you cross the two, right? And then we have all the marker effects in the database, right? We have predicted them, and then for each marker combination, you can then predict the score for that combination, and you add them up for every combination, and then you want to do that, of course, with several traits, because you're not usually interested in a single trait. So you do that for several traits that you're interested in, and then you have a selection index essentially, and that scales or weights for each trait, and it will then give you the ranking essentially that you generate that way. And it's pretty smart. So because it was developed for YAM, it actually supports dioecious plants, which means that it will only report male-female combinations.
Thank you, Lucas. Eric is asking if your seed tracking application also tracks phytosanitary test records and field inspection report.
Yes, that's a really good question, and this was actually an open source contribution by the Treatise database, who uses also Breedbase, and they have added those features to the seed lot. So if you maintain your seed lot, you want to often annotate what if the seed lots, maybe that's not your question, but the seed lots allow you to annotate the state of the seed lot, if there's any issues with infections and so on. Was that your question, or was that more related to the fields? I think it's related to the seed lots, right?
Seed lot, yeah.
Yeah. So yeah.
Also talks about field inspection reports related to the seed lot.
Yeah. So the field, I don't think we have specific reports on the field outside of a general description of the trial that you're setting up, so you could mention it there. There's no specific field for the trial that would deal with the phytosanitary records in that trial, but that's something that we could very easily add if that was needed.
Yes, it was a seed loss because we can't export materials international without this test for many crops, and it will be nice to track these details with the seed loss. That's what Eric complimented.
Yeah, exactly. Yeah, so that can be done with that extension written by the Treatise toolbox people. Yeah.
Okay. Isaac also posted that if you want to reach the main developer of the GCPC, Christine posted the email address there for Emily and others that want to. So you mentioned that has been used in several other crops other than root and tuber crops and bananas. How much has it been used from private individuals or private companies? Do you have a record of that?
Yeah, I mean, it is used. I would say it's used by a lower number in industry. It's mostly used by other large databases or university labs, actually. But definitely there are a number of companies that also use it. But usually they want me to sign an NDA agreement, so I can't talk about it.
Any other question before we close the seminar from the audience? You've been very clear, Lucas, looks like. All right. Oh, it looks like we have. Oh, Thomas said he has one, but he's taking some time to type. Go ahead, Thomas, take your time.
It'll be a huge question.
Be prepared, Lucas. So what are the, you said that things are constantly changing basically or evolving within plant breeding. What are your, you mentioned the ground penetrating radar. Is there any other things that you're working in incorporating to Breedbase?
Well, I think most recently we did this NIRS data, which is very important, and we discovered that really NIRS data and ground penetrating radar data, they are just one type of data type that is very common, and we call them high dimensionality phenotypes. And so what a lot of people want to do now is they want to take certain spectra or pictures and things like that. But also, for example, they want to do gene expression analysis, right? Or metabolics analysis. And suddenly you don't have just one data point, like plant height, it's 52 centimeters. Yeah, that's one point. But if you have a gene expression, then that's a lot of data points. That represents one measurement. And so far, our database wasn't really handling that. And the NIRS data type was really the first one that we handled that way. And we figured out that we can use a similar infrastructure to handle all the other multidimensional or high-dimensional phenotypes that way. And so we can, in the future, it will be possible to store expression data and metabolomics data and so forth. And the nice thing about that is that that data will be then also in the database, and you can really, you could potentially create very complex prediction algorithms that incorporate some of these expression data, for example. For example, you might weight things, gene regions by the amount of expression in them and things like that. So
hey, the big question came from Thomas. Okay, he's wondering how much crossover between computer science and plant breeding is necessary to design this type of tools, and are the people developing these tools experienced in breeding and as such knowledgeable what they need, or is there a back and forth between people who are primarily breeders and people who are primary programmers? and what they need out as of a tool and what tools are feasible to create.
Yeah, I mean, it's a mixture of things, right? So at Breedbase here at Boyce Thompson Institute, we have in the group we have trained plant breeders, we have trained software engineers and so forth, biologists. But I think there always has to be a back and forth between the user and the people who create the software. I mean, that's just clear that if you create some software in isolation, it just won't be useful, right? And what are the needs and so forth. And so I think we have tried to listen a lot to our users, especially in these African, crops, the RTB users, as well as other users that use the database here, such as the Treatise group that also provides a lot of code, actually, which is extremely nice. So sometimes they provide entire tools, as well as the other users, that here in university labs that use it. They have been extremely valuable for feedback. So it's always a back and forth. You cannot create a tool in isolation. Nobody knows everything. So it really is a big collaboration.
How about the incorporation of on-farm trial data? I think that also is a response to some of the demands from these groups.
Exactly. I haven't talked about that, but we're working on that. We're working with the CleanUp group to interface with their tool, so that you can then, once you have, so we will always just, I think that's the current thinking. You design your trial, of course, on CleanUp and use their infrastructure. When your experiment is done, you can transfer the results to Breedbase, and the breeder can then log into Breedbase, and it has all the information, also the farm-based trial information in the database.
Good. Well, thank you so much, Lucas. I just want to also thank Tiago and Brandon for their help in running this seminar series. We have the last seminars of this series next Thursday by Dr. Hamid Kasai from Finland. He's going to talk about fava bean breeding and genomics. Same time here, November 9th at 11 a.m. Thank you so much. Thank you, Lucas. And you guys have a great weekend, and we'll see you next Thursday.
Thank you so much. Have a good one.
Thank you.
See you next week.
Bye-bye.